Viewers, welcome back to my channel. Here I am back with a new another video, and today's session we are discussing about the ribosomes in the chapter 2 zoology CBZ second year CBZ fourth sem. So ribosomes, where the ribosomes are present. These are present in the cytoplasm, or also it is sometimes attached to the surface of the endoplasmic reticulum, as we know before. So, and then the ribosomes are also known as basophilic granules. Why it is known as the basophilic granules means, basophilic means small or minute. Hence we call ribosomes as the basophilic granules. Here, Robinson and Brown in 1953 in bean's root or in plant cell firstly discovered the ribosomes. Here the ribosomes means the protein plus RNA. So he observed the RNA and the protein, and in 1955 it is rediscovered by George Pellet in animal cell. He also observed the RNA and the proteins, and then the Robert gave the term ribosomes. Here the main are Robinson and Brown in 1953 they discovered in the plant cell in bean's root, but in 1955 rediscovered by George Pellet in animal cell, and then the term ribosome is given by the Robert. So clear? And then the by-centrifugating animal tissue and detected RNA in the animal tissue. Here the ribonucleic acid called as ribonucleoprotein. Firstly, the ribosome's name is the ribonucleoprotein, and then it is further named by the Robert. Then the name ribosomes came. This is the history. But in both plants and animal kingdom, but also in bacteria, prokaryotics and eukaryotics, we gonna see or we gonna observe ribosomes. Here next came to the distribution. In prokaryotes, protein factories. Ribosomes are known as the protein factories. Another name for the ribosome is the protein factories. Here the proteins or ribosomes are present in the cytoplasm of the cell, or also known as the free ribosomes, because they were not attached to any of the other cell organelles. For example, as I said before, it is sometimes attached to the endoplasmic reticulum. Here in prokaryotics, they are not attached to any of the other cell organelles. Hence, they are known as the free ribosomes. But in eukaryotic, it is opposite to the prokaryotic. The ribosomes are attached to the surface of endoplasmic reticulum, and they are not free in nature. And then, how ribosomes are made, or what is the composition of the ribosomes? Here, the mixture of protein plus RNA gives rise to the ribosomes. Here, the mixture of protein and RNA, when they combines, they form the ribosomes. And then, where it is producing. In eukaryotes, nucleus has a round structure called nucleolus. In nucleus, the nucleolus is present, as we know all. Then the nucleolus is a site of the synthesis of ribosome production. In nucleolus, particularly in nucleolus of the nucleus, in eukaryotes, synthesis of ribosomes or the production of ribosomes takes place. Here the ribosomes are of two types, 70S and 80S, or ribosomes has the two units in ribosomes, the larger unit and the smaller unit. The larger unit is dome shaped and the smaller unit is base shaped. Then the component, here the Mg plus 2 or 2 plus magnesium is present. If magnesium is present in larger amount or the concentration of magnesium is high, the two subunits get attached. If the magnesium concentration is low, they do not attach, instead of they detach. So when the magnesium concentration is high, the two units, larger and the smaller unit, get attached together, and when the magnesium concentration is low, the larger and the smaller unit get detached. And when the magnesium concentration is decreases, the production of ribosome level also decreases. So there is a main the magnesium plays a main role in the production of ribosomes. And then move on to the in prokaryotes. Prokaryotes are also known as the simple cells. Here the 70s ribosomes, or why we call it as a 70s ribosomes, means the cell size is reduced as compared to the eukaryotes, because these are the simple cells. Hence the size is reduced when compared to the eukaryotes. Hence the molecular weight is also reduced in prokaryotes, and the ribosomes are the 70s. And but in eukaryotes, the molecular weight and the size is more as compared to the prokaryotes. Hence the ribosome is 80s in eukaryotes. The main what is the S here? Here S is the Swedberg unit or the sedimentation coefficient. What is the sedimentation? It is the centrifugation, or other term is sedimentation. Here the eukaryotes are known as the complex cell. Cell size is large, hence 80s. Ribosomes are large in size. In prokaryotic, cell size is reduced, hence 70s. Molecular weight also small. Here, as I said before, what is S? Its unit nothing but 70 Swedberg unit, or extraction of ribosomes through the centrifugation or sedimentation is called as the Swedberg unit or sedimentation coefficient. So thank you for watching. Please do share, like and subscribe to my channel. Stay tuned. Thank you.